Hello everyone, my name is Muleya and today I will present my findings on the speciation and bioaccessibility of selenium biofortified crops of dietary importance from Malawi. This is the outline of my presentation. I will first talk about the importance of selenium biofortified crops and explain why speciation and bioaccessibility of such crops is an important consideration. I will talk about the approach we took and show you some of our findings. I will end with a conclusion and future outlook. First, the importance of selenium biofortification. Selenium is an essential micronutrient incorporated in at least 25 selenoproteins in the human body. Selenoproteins play a critical role in several metabolic processes in the body. Selenium is required in very small amounts, with recommended dietary intakes ranging between 30 to 85 micrograms per day. However, despite such low requirements, suboptimal intakes of selenium are common in many parts of the world where soils are largely selenium deficient. Uptake of selenium from soils to crops is an important entry point of selenium into the food system. And agronomic biofortification, which is achieved through the application of selenium-enriched fertilizers, has been found to be an effective strategy to increase selenium supply in the food systems of regions where it is deficient. For biofortification to be successful, the applied selenium must be bioaccessible or bioavailable for it to achieve the intended effects in the human body. It is therefore important to understand the bioaccessibility of these crops. Bioaccessibility would depend on the speciation of selenium in the edible portions of the crops. Speciation refers to the chemical forms in which selenium exists in a particular crop. Selenium can exist as inorganic and organic compounds, and it has been established that organic compounds are more bioaccessible than inorganic compounds. Bioaccessibility will also depend on the crop matrix. For example, there could be differences between cereals and legumes, or differences induced as a result of processing. The total concentration of selenium in the edible portions of biofortified crops is derived from selenium native to the soil and from the fertilizer. It has not been established whether there are differences in the speciation and bioaccessibility of the two different sources of selenium. In the next slides, I will discuss the approach we took to investigate these aspects using selenium biofortified crops of dietary importance in Malawi. We obtained maize, cowpea, and groundnut samples from an isotopically labeled field trial at Chiteze Research Center in Malawi. The crops were grown under 11 different conservation agriculture management strategies. Isotopically labeled selenium in the form of sodium selenate was added to crops at a rate of 20 grams per hectare. The edible portions of the crops were harvested and analyzed for total selenium concentrations using ICP-MS, in which the isotopes selenium-77 and selenium-80 were monitored in order to discriminate between native selenium from soil and applied selenium from fertilizer. Speciation was done using an HPLC coupled to ICP-MS, and finally, selenium bioaccessibility was determined after subjecting the unprocessed samples through an in vitro gastrointestinal digestion model. Soluble selenium derived from the in vitro digestion was considered as bioaccessible. In this experiment, we did not investigate the effect of processing. I will show some of our findings. This graph shows the total selenium concentration. On the x-axis is the maize, cowpea, and groundnut crops grown under different conservation agriculture management strategies, and on the y-axis is the selenium concentration in micrograms per kg dry matter. The red columns represent native selenium from soil, while the blue columns represent selenium from applied fertilizer. From this graph, we can see that the selenium derived from soil is many magnitudes lower than the selenium from applied fertilizer, indicating that indeed, agronomic biofortification of crops is an effective strategy to increase selenium concentration. We can also see that legumes, in particular groundnuts, accumulated more selenium than maize. This graph shows a typical chromatogram from the speciation analysis of maize grain. In the x-axis is the retention time, and in the y-axis is the intensity of selenium. The red line shows the peaks of the native selenium from soil, while the blue line shows the peaks of the applied selenium from fertilizer. We can see that both profiles are similar, differing only in magnitude, with one major peak of selenomethionine and two minor peaks of selenocysteine and selenate. This proves that both sources of selenium are converted into the same species. Similar profiles were also obtained for groundnut and cowpea. This graph shows the proportion of selenium species in relation to the total extracted selenium. More than 90% of the extracted selenium was in an organic form, indicating that the crops were efficient at converting inorganic selenium applied as selenate into organic selenium. 3 organic species were identified: selenomethionine in blue, selenomethylcysteine in red, and selenocysteine in gray. In all cases, selenomethionine was the dominant organic species, constituting at least 85% of the extracted selenium in maize and groundnut. An interesting finding was the occurrence of selenomethylcysteine in cowpea, constituting about 30% of the total extracted selenium. Inorganic species, selenite and selenate, were also identified, but were in trace amounts. This table shows the bioaccessibility of applied selenium as a percentage in the first column and in concentration terms in the second column. No differences in percent bioaccessibility were observed for all crops, with an average bioaccessibility of around 75%. This is probably because of the dominance of organic species, which are highly bioaccessible. However, in concentration terms, groundnut provides more bioaccessible selenium than both maize and cowpea because of its initial higher selenium concentration. Nonetheless, bioaccessible selenium concentrations in maize and cowpea are still high enough to substantially increase selenium in the Malawian food system. To conclude, we have shown that selenium biofortification of staple crops is a viable strategy leading to bioaccumulation of highly bioaccessible organic selenium species. The accumulation of selenomethylcysteine in cowpea, one of the most potent anti-carcinogenic selenium species, could lead to more health benefits that go beyond improving selenium status. Some more insight in this aspect would be valuable and could guide an understanding of how functional products could be engineered at the agricultural level. Finally, the effect of typical processing methods on speciation and bioaccessibility needs to be established, as bioaccessibility is also influenced by processing. Thank you.